A single strand of white fuzz on a morning glory seed, seemingly insignificant, yet it cracked open a mystery that has haunted chemists since the 1930s. Could the original source of LSD-like compounds be hiding in plain sight? For nearly 90 years, chemists and botanists have wondered, what fungus was producing the powerful ergot alkaloids found in Mexican morning glories? These compounds, chemically similar to the psychoactive ingredients behind LSD, hinted at a hidden symbiotic partner, one that Albert Hofmann himself suspected but couldn't isolate. Now that mystery has finally been solved. The implications go far beyond a historical curiosity. They touch the future of medicine, biotechnology, and how we understand the delicate partnerships between plants and fungi. They also raise new questions. How many other powerful biochemical relationships are we still blind to? What other evolutionary alliances lie hidden within nature's most unassuming organisms? The discovery also challenges assumptions in mycology and botany that have remained largely untested for decades. It's a reminder that plant-fungal relationships are more intricate than previously thought, and that groundbreaking insights can still come from humble species like the morning glory. In a study published on April 22, 2025, in the journal Mycologia, Corinne Hazel, an undergraduate student at West Virginia University, made a discovery that eluded some of the greatest minds in pharmacology. A junior majoring in environmental microbiology, Hazel had joined a research group led by Professor Daniel Panaccione, a plant and soil sciences expert in WVU's Davis College of Agriculture and Natural Resources. Their focus, studying the source of ergot alkaloids, psychoactive compounds, inside Ipomoea tricolor, also known as heavenly blue morning glory. Decades earlier, Albert Hofmann, the Swiss chemist who famously synthesized LSD in 1938, had encountered these same alkaloids in Mexican morning glories. He suspected they weren't produced by the plant itself, but by a fungus living symbiotically within the seeds. But no one had ever managed to isolate that fungus. It became a decades-long dead end in psychedelic pharmacology. Hazel wasn't even trying to solve that particular mystery when she made the discovery. It happened almost accidentally. We had a ton of plants lying around, she recalled. We noticed a little bit of fuzz in the seed coat. That was our fungus. What looked like ordinary seed fuzz turned out to be the long-lost producer of lysergic acid derivatives. With a research grant, she secured herself, no small feat for an undergraduate. Hazel extracted DNA, sequenced the genome, and confirmed the discovery. A new fungal species with the biochemical machinery to synthesize ergot alkaloids. It was a moment that rewrote a chapter of both fungal biology and pharmacology. And it underscored something else too, how much power there is in observation. In a world increasingly reliant on high-throughput screening and automated analysis, it was a simple visual cue, noticed by human eyes, that revealed the key. Her work also illustrates how academia can still serve as fertile ground for original discovery, even for undergraduates. The freedom to explore, to ask unusual questions, and to examine discarded clues is sometimes all it takes to break open a mystery that has remained sealed for generations. The research team named the fungus Periglandula clandestina, a nod to its long-hidden, nearly undetectable presence. As it turns out, this seed-dwelling fungus had successfully evaded discovery for nearly a century due to its cryptic lifestyle. Unlike other fungi that grow visibly on leaves or stems, P. clandestina hides inside the seed coat itself, invisible unless you're looking very closely. And it was that exact fuzz, barely noticeable, that finally gave it away. Once isolated, chemical analysis confirmed the fungus was actively producing lysergic acid derivatives, specifically compounds such as lysergic acid hydroxyethylamide, LAH, which are structurally similar to LSD and have both psychoactive and medicinal properties. These compounds belong to a broader family of ergot alkaloids, many of which are capable of interacting with human serotonin receptors, making them powerful tools and potential risks in neurological medicine. This is the first time anyone has isolated the fungus from morning glory seeds that's actually making these compounds, Panaccione said in a university statement. It closes a major gap in the scientific record. The implications are significant. Ergot alkaloids are used in medicine for treating migraines, Parkinson's disease, dementia, and in some cases as vasoconstrictors during childbirth to prevent hemorrhage. But they're also highly toxic in large doses. The same family of compounds has caused mass poisonings throughout history, most infamously in the Middle Ages, when people consuming contaminated rye suffered from convulsions and hallucinations in outbreaks known as St. Anthony's Fire. As Panaccione noted, many things are toxic, but if you administer them in the right dosage or modify them, they can be useful pharmaceuticals. By studying them, we may be able to figure out ways to bypass the side effects. And there's another benefit. Fungi like P. clandestina could one day serve as biofactories, natural producers of medically relevant molecules that would otherwise require complex and expensive chemical synthesis. The fungus might even be genetically modified to churn out safer, more targeted compounds for pharmaceutical use. Even more exciting, this discovery may be just the beginning. There are over 600 species of morning glories worldwide, many of them used in traditional medicine or ornamental gardening. Each may harbor their own cryptic fungi with unique biochemical talents. Hazel's next challenge is attempting to culture P. clandestina in the lab. It's a slow grower, difficult to cultivate under standard conditions, but unlocking that step could offer researchers a stable, reproducible way to study its chemical output. It would also allow the development of high-throughput screening methods to test for new alkaloids with untapped therapeutic value. And there's reason to believe that P. clandestina is just one of many fungal species engaging in these complex, often hidden chemical dialogues with their host plants. Fungal biodiversity remains one of the least understood aspects of Earth's ecosystems, and discoveries like this one highlight how much we've yet to uncover. Some scientists even speculate that the vast majority of plant bioactive compounds may involve fungal partners, meaning we've only been studying half the equation all along. The discovery of Periglandula clandestina does more than solve a historical riddle. It creates an entirely new platform for both pharmaceutical exploration and agricultural safety. In pharmaceutical terms, isolating the fungus allows for direct manipulation of its genes, opening new possibilities for drug synthesis using fungal biotechnology. Instead of synthesizing complex compounds like ergot alkaloids through multi-step chemical reactions, scientists may eventually use P. clandestina itself, or genetically modified strains derived from it, to mass-produce medically useful molecules with fewer side effects. This isn't just speculative. Research in fungal synthetic biology has already yielded breakthroughs in antibiotic and immunosuppressant production. Now that the full genome of P. clandestina is mapped, scientists have the blueprint for manipulating its alkaloid-producing genes, either to amplify yields or to silence toxic byproducts. On the agricultural front, understanding how fungi like P. clandestina form symbiotic relationships with host plants could help prevent toxic contamination in other crops. Ergot poisoning remains a threat to grazing livestock and human grain consumption in some parts of the world. By tracing the fungal pathways, scientists could design better safeguards against future outbreaks, whether by breeding resistant crops or designing safer symbioses. Hazel's work also highlights a powerful shift in scientific discovery, the rise of student-led research breakthroughs. In her words, "I'm lucky to have stumbled into this opportunity. People have been looking for this fungus for years, and one day I look in the right place, And there it is. It's a reminder that major scientific advances don't always come from billion-dollar labs. Sometimes they start with a curious student, a patch of morning glories and a fuzzy seed. And perhaps more than anything, it reminds us how much we still have to learn from nature's hidden partnerships. Every overlooked seed coat, every dusty petal might be the key to a future medicine or the solution to a modern agricultural threat. From a nearly invisible thread of fuzz on a garden plant, a young scientist unveiled one of the longest-standing mysteries in psychedelic pharmacology. The elusive fungus Albert Hofmann once theorized now has a name, a genome, and a future in medicine. It took 90 years and a fresh pair of eyes to find it, so what other secrets might still be living quietly inside seeds, stones, or spores, waiting for someone to notice? Subscribe now for real science, updated daily from peer-reviewed studies across the globe. Stay curious, stay informed, and discover the next story hidden in plain sight.